Alright, now let's cover hexokinase versus glucokinase. Now do you remember the function of a kinase enzyme? There you go, the kinase enzymes transfer a high energy phosphate group from ATP to a substrate. Here we see a cell and a molecule of glucose outside the cell. And in this state, it's free to enter and leave the cell as it pleases. But once glucose enters the cell, it usually meets with hexokinase or glucokinase enzyme. And this enzyme will add a phosphate group to the glucose molecule. And if you think about it, this is the first step in glycolysis, the phosphorylation of glucose to create glucose 6-phosphate. And this reaction uses 1 ATP. Now why is the phosphate group added to the glucose here? Well, in addition to preparing the glucose for the next step of glycolysis, it also gives the glucose a negative charge, which prevents it from passing through the plasma membrane, effectively trapping the glucose inside the cell. Now this reaction can be catalyzed by either hexokinase or glucokinase. And the difference between these two is in their location of action as well as their enzyme kinetics. Now you're going to talk a lot more about enzyme kinetics in the pharmacology section, especially Km and Vmax, which you can see here on the graph. And for now, we're going to focus on the kinetics of these two specific enzymes, hexokinase and glucokinase. Now hexokinase is found in almost every cell type in the body, except for liver cells as well as pancreatic beta cells. Now hexokinase has a high affinity for glucose, but it has a low maximum reaction rate. This means that the concentration of glucose needed for this enzyme to be used is relatively low since it has a high affinity for glucose, but the speed in which the reaction occurs is fairly slow because it has a low Vmax. Hexokinase is not affected by insulin, but it does receive negative feedback from high amounts of glucose 6-phosphate. So basically, when the cell has low amounts of glucose 6-phosphate, hexokinase will catalyze any glucose available to glucose 6-phosphate to store it within the tissue. However, if there's a large amount of glucose in the blood, such as after a meal, hexokinase won't be able to keep up since there's a low capacity or low Vmax. Next up, we have glucokinase. And glucokinase comes into play after meals, when there are high glucose loads in the bloodstream. And where is glucokinase found in the body? Well, glucokinase is found only in the liver and pancreatic beta cells, which is essentially the exact opposite of hexokinase. Now this enzyme has a low affinity for glucose, but a high capacity or reaction rate. This means that glucokinase is not able to be active unless there's a lot of glucose available. But if you have a lot of glucose available, it can act very quickly to phosphorylate it. Also, glucokinase has no negative feedback inhibition from glucose 6-phosphate. So as long as there's lots of glucose in the bloodstream, glucokinase will keep phosphorylating the glucose inside the cell. You can think of glucokinase as kind of being a buffer system, because after you eat a large meal, the high glucose levels will activate beta cells in the pancreas. These pancreatic beta cells will then release insulin, further activating glucokinase in the liver so that glucose can be removed from the bloodstream and stored as glycogen. Now what's another way to think of hexokinase and glucokinase? Well, I like to think of these two enzymes like the tortoise and the hare. Hexokinase is the tortoise because it acts in a slow and steady pace, and glucokinase here is the hare, speeding up and slowing down depending on the amount of glucose available in the blood. I also want to touch on the concept of maturity onset diabetes of the young, or MODY. Now you're going to learn more about the disease diabetes as a whole in the endocrine section, but in a nutshell, diabetes results in an increase in glucose in the bloodstream. Now there are many different forms of diabetes, and these include type 1 diabetes, type 2 diabetes, as well as MODY, which we're talking about here. Now maturity onset diabetes of the young is a group of monogenic diseases, meaning that only one mutation in one of a few genes causes diabetes, versus the other forms of diabetes which have multiple genetic or non-genetic causes. Now one of the mutations in MODY involves the glucokinase gene, and this mutation will functionally alter the glucokinase enzyme, producing this form of diabetes. Now why do you think this is? Now remember, glucokinase is induced by insulin when there's high levels of glucose in the bloodstream, as I've tried to depict here. Now in MODY, there's a point mutation in the glucokinase enzyme that causes it to malfunction, and in this case, insulin will not be able to activate glucokinase as it normally would. This results in increased levels of glucose in the bloodstream, because the glucose would no longer be trapped inside the cell. Now it's important to note that a hexokinase mutation has not been associated with MODY, just a glucokinase mutation. Well, alright, let's take a shot at a flash quiz. A 65-year-old man with type 2 diabetes injects insulin before lunch. How does the insulin affect the action of hexokinase and glucokinase? Well, hopefully you remember that insulin induces the enzyme glucokinase, but doesn't affect hexokinase. This is because hexokinase is always active, while glucokinase must be stimulated by insulin when glucose levels are high to create more glucose 6-phosphate and to trap the glucose inside the cell. Well, alright, that's going to wrap up hexokinase versus glucokinase. See you in the next video.